Welcome to our webinar series for professionals.
I'm Anne-Catherine, co-founder of Nordic Laboratories and DNA Life, and today I'm joining with Helen Gautschi, our geneticist at DNA Life, and she's gonna discuss more around Growbaby, treatment strategies, biochemistry, genetics, etc., around healthy pregnancies and outcomes. We are recording this, so we'll send out the recording. Helen will share her slides, so we'll send out the slides with the recording. Chat in the chat box, ask questions, answer each other's questions. I'll take note of those that can't be answered, and then we'll do the Q&A at the end of Helen's presentation. I'll unmute myself, Helen, and let you, I'm sorry, Then, of course, Helen, you are our specialty geneticist in DNA Life, and you're an excellent brain, and I love your education, and I just love working with you. Some of you have met Helen before as well, so I kind of like, oh, here comes Helen. But Helen, share your slides on your screen. I'll mute myself because you're the star of the show.
Thank you for having me, Anne-Catherine. And I see some familiar names and faces, so it's lovely to be here again. Thank you. I'm going to just share this, and then, so thank you everyone for having me. And today we're going to be speaking about it is a niche test, as Anne-Catherine says, but I think has much more applicability than we give it credit for. So let's dive into Growbaby, and please, if you have any questions, I'd be happy to answer them at the end. So getting into it, today we're going to be discussing the Growbaby test, applications of the test within various areas of maternal health, from fertility through to maternal and fetal health, birth outcomes, and then also considering transgenerational health thereafter. So why are we here? What are we talking about? Maternal health, why it's so important. There have been efforts to improve maternal health, and it has been a priority for decades worldwide. Despite, however, significant advancements, it remains a major public health concern, and it was the cause of over 1.28 million years lived with disability, and this was a 2019 statistic for this, and this was globally what they found there. In terms of maternal health disorders, it contributes towards increased risk for premature or poor birth outcomes from premature birth, small or large for gestational age, and the consequences thereof, as well as pregnancy loss. And then also increased risk for long-term health disorders, both in the mother, leading to increased risk for, if there's gestational diabetes, increased risk for things like type 2 diabetes and other cardiometabolic disorders for the mom long-term, and also in her children long-term. And so we're wanting to look at how we can address both those short and long-term outcomes. And then compounding this problem that's been coming to light more recently is this increased barrier in fertility and the rise in assisted reproductive technologies, such as your in vitro fertilization or your IVF. And what we also know, as what evidence suggests, is that maternal as well as paternal dietary intake and environmental exposures pre-conception are really, really important in playing a role in fertility, as well as pregnancy and the health of that pregnancy and offspring outcomes. And what we definitely know is that maternal diet and environmental exposures throughout that perinatal time period are integral to positive birth outcomes and transgenerational health. And this is where I've got some articles here that when you get the slides, they're lovely articles to go and read. So please go have a look at those. So cue the idea of what can we do about this concern. And what we see now is that preconception and the perinatal time period provide a unique opportunity to establish the foundations for optimum health and development across the lifespan. targeted interventions using insights from genetics, plus your biochemistry and your functional tests that pair with the genetics, can optimize the health of the mother, supporting a healthier pregnancy and resulting in transgenerational health and resilience. This is where Growbaby was born, or the Growbaby DNA test was born, around 2018 or so, and 2019. And I want to just really emphasize where this test would be useful. So it is really integral and really important to consider any woman coming into your practice who is of reproductive age. This test will be useful for them. So any woman who plans to conceive, but remember, not all pregnancies are planned. So for optimizing health during that pregnancy and beyond, if you can do the test before conception, that's really such a jewel of a bit of information to be able to use this information to really lay the foundation for health going forward. It's also really useful for the couple, but the mom to be considering or attempting IVF, or looking at, or who's struggling with fertility, so that subfertility space, and using any other kind of assisted reproductive technologies for conception. This is a really good test to consider, because there's so much in here that you can support, and you can understand the biochemistry and the biological pathways that might be lacking or weakened because of genetic variation. And if you can support that from early on and get those eggs really healthy, when you get the eggs, they're not just eggs, they're healthy eggs. And that's a really good place to start off with as well. So in that fertility space, a lot that you can do with over here as well. Of course, we can't forget women who are already pregnant, and the study that was actually that was going on in the US by our Growbaby team, Dr. Emily Ribbaum and Drs. Leslie Stone. They actually had their cohort of women who were already pregnant, high risk pregnancies, and they got really good results from there. So really not forgetting, if you're already pregnant, there's so much you can still do and support with this test. And then any woman who has experienced previous complications with regards to conception or pregnancy, including pregnancy loss, preterm birth, preeclampsia, gestational diabetes, and looking to have a baby or wanting to conceive again, this is really the area where you can go in and support those biological pathways as much as possible to improve health going forward. So what does the test really consider, and what are we looking at within the Growbaby test? So we look at the research and where there are gene, environment, phenotype interactions, where the evidence is really strong and where it is clinically useful. And this is where we are able to then report on specific birth phenotypes, outcomes, and conditions. Under the birth phenotypes, we look at how can we, or look at the genetic variants that are associated with small or large for gestational age, risk for preterm birth, and then a stress dysregulation phenotype in both the mom and the baby. Birth outcomes are really more on the mom's side, so things like gestational diabetes, preeclampsia, your gestational hypertension or pregnancy-induced hypertension, miscarriage and recurrent pregnancy loss. And then in terms of birth conditions, we look at variants associated with neural tube defects, and how, based on your environment, can we offset those weaknesses to promote an optimum pregnancy and optimal birth outcomes. And so this is where we become very targeted with the interventions. It's not just looking at your baseline support that is required during pregnancy, which is already a very special time period, but we're going far beyond that with our interventions and the recommendations. And so this is where I'm going to jump in a few examples. At the training in the UK, that intimate afternoon that I'm looking forward to, we will go through more examples, discuss more biological pathways, more scenarios and case studies. But this is where I want to highlight one or two where the evidence is so good, and it really gives so much insight into what this test is about. So this is actually a Venn diagram that was developed in partnership with Emily Ridbaum. She's great with these insights and developing these kinds of pictures. And so really highlighting the importance of looking at the strong overlap between genes and phenotype and biological pathways and phenotype, that it's not just one gene that leads to a specific phenotype, and it's just as one biological pathway. But if you see that variant is present, how it can affect multiple phenotypes. So there's a strong overlap. And something just always to keep in mind, that when you can support one variant or support one gene, if you have a variant there, you're actually supporting and optimizing health across many phenotypes. And this is really, I think, very integral when looking at Growbaby. But and something that's actually with all of our tests is that we have that when supporting the biological pathway, we can support many phenotypes and actually support health and resilience long term. So just highlighting the pathways that we do report on in the Growbaby test, from lipid metabolism, down inflammation, your detoxification, methylation, your monoamine oxidase metabolism, and your neurotrophic pathway, which is involved in that stress dysregulation phenotype, progesterone metabolism and the receptors over there, melatonin, which I'm going to get into a bit more, insulin sensitivity, secretion and metabolism, and your vitamin D requirements. And how we report on it in Growbaby is where you carry variants that lead to an altered enzyme function, possibly weakening that specific pathway, you will get a little up arrow saying elevated support required in that area, and you know exactly where that targeted intervention needs to take place. So I'm going to start off with a scenario of looking at fertility. So we're starting off early, and how we can support fertility through understanding insights from the Growbaby test. And just handpicking a specific area here is with methylation. So we see it's not really highlighted here so much, but you'll see some MTHFR, all of the methylation genes popping up here and there, and how it relates to fertility. So why fertility? I mentioned it briefly earlier, but it is becoming a growing concern also because couples are considering having children later on once they're more established. And so we see prevalence in terms of infertility or subfertility ranging from 8 to 12%. So 1 in 8 couples eventually end up seeking medical treatment after failing to conceive for 1 year. And that leads to this increased use of your assisted reproductive technologies. And when we're looking at that increased use, we again want to make sure that everything is supported as early on from a biochemical and a nutritional pathway point of view. And this can really save a lot of heartache and just make sure that everything is as healthy and that foundation is set as early on as possible. And so when we're looking at the reasons or some of the reasons for fertility issues, they can be due to epigenetic changes, obviously increasing age, there's increasing oxidative stress, environmental stresses, reproductive disorders such as your PCOS or your endometriosis also contribute towards this infertility or subfertility space. And how does methylation fit into all of this? Because it's a very big area that we can dive into, and I'll definitely speak about more of these areas at the longer in-person training, but really highlighting the importance of methylation here is, and just going through the pathway first off. So we've got 2 scenarios. We've got the one that you can see on top is everything going right and leading to a nice, healthy, normal pregnancy, everything just flowing as it should. And the bottom scenario is where we don't get the adequate nutrients, where there might be abnormalities in enzyme function and in the pathways that's leading to a decrease of the metabolites that we want and maybe an increase of the metabolites that we don't want. So how does this work? And just to go through the steps as a reminder that I'll look at the one where if we have optimum and I'll keep on touching on the one where things go wrong. So how does methylation fit in here and why it's so important? So we have our nutrients, and this is the example where we've got our dietary folate coming in. And there can be little blocks at different points, or these rate limiting steps that occur. And if at these specific points, things aren't working well, you have a genetic variation that's leading to an enzyme kind of malfunctioning, or you're not getting in the right nutrients, that adds to those blocks and a difficulty in creating the nutrients that we need to support fertility and a healthy pregnancy. So we start off looking at something like your dietary folate, your other nutrients coming in here as well, your B12, your B9, your B6, and so on. But your folate here coming in through tetrahydrofolate, and that would be your first rate limiting step. So if you're getting in a supplement such as your folic acid, where it needs to be converted to tetrahydrofolate, that's already a step. And that's an enzyme that needs to do that. So if you've got problems in that, or if you've got absorption issues or anything like that that might be occurring, that's the first step where there's this kind of block in how much of this tetrahydrofolate you're getting in. It then gets converted to your 5,10-methyl tetrahydrofolate, and then through this enzyme, your MTHFR, your methylene tetrahydrofolate reductase, reducing that 5,10-methyl tetrahydrofolate to your 5-methyl tetrahydrofolate, is this important methyl donor that can then be used to, with homocysteine, create methionine. So think of a baker man taking this ingredient. We need the magic ingredient 5-MTHF, and we take homocysteine and we put them together and we make methionine. So if we don't have our MTHFR enzyme working properly, we again have this rate limiting step, or this block occurring, where we aren't able to convert homocysteine sufficiently to methionine, leading to almost an imbalance occurring in that space, where we might have too high homocysteine levels and too low methionine levels, which then leads to the inability to then create our SAM, our S-adenosine methionine, which is our universal methyl donor that can then be attached to our DNA, to hormones, and to create our phospholipids, our neurotransmitters, and to create our DNA, and to kind of switch on and switch off our genes appropriately. So this is where that next rate limiting step occurs. Do we have enough B12? Is our methionine synthase enzyme working? And so on. So we test a lot of genes in this pathway that are related to the functioning of methylation and the creation of our SAMe, and looking at the levels or the potential levels that could be created between our homocysteine and our methionine. And is that healthy, or is it not in balance? And so if it's all healthy, we end up having this normal pregnancy outcome. However, if it is not in balance, there is an increased risk for altered regulation of gene expression of these vital genes involved in placental development, leading to adverse pregnancy outcomes, and then increased risk for non-communicable diseases in the offspring later on. So how this works, I just want to dive a little bit more into this area around fertility, and this is actually looking long-term, and I'll get to it in the next page and the next slide. But how methylation works when we're talking epigenetics is we've had the methylation pathway explained here. We have our SAMe that is then created with these nutrients. We then get these little these building blocks that then create this methyl group on SAMe. It's this little present that needs to be attached to our DNA, and it's like this tag that gets attached. And the more tags you attach to your DNA, the more methylated that part of the DNA is. And if you almost think of it as like reading a book, if you've got lots of kind of cellophane or something covering up that part of the book, you can't read that page very well. And so if you're wanting to copy that information, you can't copy it because you can't see what's there. And so that part of the gene won't be expressed that easily because it's covered up. And that's the same with your methyl groups. There are some places in your genes that you want to cover up, and there are other places in your genes that you don't want to cover up. And your body is very clever at knowing where to cover up and where not to. But this is also determined on the methyl donors that you have available to you. And so for normal methylation to occur, your body needs those methyl donors in the right amounts, needs to make sure that redox balance is there, so not too much oxidative stress either occurring, and not too much inflammation, just the right amount, and all the signaling happening properly so that your methyl groups attach to the parts of your DNA that need attaching, and so that expression can happen in the right amounts. And so when genes need to be turned on, they can be turned on, and when they need to be turned off, they can also be turned off. And that's going into this area of epigenetics. And so it's also, epigenetics is governed by what we put in from our environment, plus how our genotype, what our genotype is telling our body to do. Do we have genes that are encoding for very nicely functioning enzymes, or do we have genes that are coding for enzymes that need more support from our environment? And this is really important when it comes to fertility, actually long term. So the first part of methylation and fertility that I am touching on is actually speaking about what happens from a transgenerational perspective. What happens in the mother can affect what happens in children and in generations to come, and how and supporting their fertility actually in generations to come, and that transgenerational health. So that's the first thing, and there's very interesting research going into that part of it, where what we can do for our patients now can affect their offspring and their future generations' health. So very important in that perspective. And just an example here, I mean, these are mice, but there's another study on the next slide that's around humans. But again, highlighting the importance of methyl donors and remembering that we have power over our genes. So this is the example. I'm sure many of you are familiar with this. This is the agouti mouse. We've got these two mice had the exact same genotype. And so what they did in this study is they took agouti mice, all these big, large yellow mice that tend to have metabolic disorders. One set they gave, and they all carry this agouti gene. So the one set they gave a mouse diet with a lot of BPA exposure as well. And then the other set in a different cage, they gave BPA as the exposure, but then they gave, instead of a mouse diet, a normal mouse diet, they gave a methyl-rich diet. And those mice were pregnant, they had babies, and the mice who had the methyl-rich diet, even though they had the BPA exposure, gave birth to these small brown little mice. So what happened? The gene that encodes the agouti protein that leads to this agouti phenotype, this metabolic phenotype, was silenced because of the methyl donors. Whereas in the mice who did not have sufficient methyl donors, they gave birth to big fat yellow mice. And so we can see the importance of transgenerational health occurring in this instance. And we see it through other studies and longitudinal studies, cohort studies, where we see the importance of exposure and nutrition from a transgenerational health perspective, actually affecting metabolic traits, metabolic health, and fertility in the generations to come. And so this is an example where it's a nice study looking at maternal intake of methyl group donors affecting DNA methylation of metabolic genes in infants. And some really interesting studies coming out on the metabolomics of infants when we're looking at what the mother was given during pregnancy. So again, just emphasizing the importance of methylation and epigenetic modifications in pregnancy, and how that transgenerational health actually occurs over many generations, from even looking at maternal stress as an example during pregnancy, and DNA methylation of the third generation, and how that still continues to affect the germ cells of that third generation. So very important, considering what we can do now to impact fertility of generations to come. And then looking much more immediate and close at hand, what can we do for women right now who are looking at improving their fertility? And here we're looking again and considering methylation and remembering that homocysteine as almost an oxidative stress marker. If it's too high, it can cause a lot of oxidative stress and inflammation, and it can lead to an increased risk of subfertility. So in this study, which was actually done quite recently, we're looking at the association of homocysteine, and especially in this area of assisted reproductive technology with using IVF and IVF outcomes. These women were stratified by MTHFR genotype, and they looked at homocysteine genotype and birth and fertility outcomes, fertility success. And so what they were able to see from this is, from the study, they saw that homocysteine is highly negatively associated with clinical pregnancy and implantation rate. And this is especially seen in women carrying the TT genotype. So if you are an MTHFR TT genotype carrier, you come out with having a high homocysteine level, there is an increased risk of poor fertility. Whereas if the homocysteine, so negatively associated, so if the homocysteine is lower and you are TT genotype, and that's probably because you're getting insufficient methyl donors, then your chances of fertility are much greater. So that's that cup half full analogy where we talk about if you have the variant, but you put in the right environment, you can get a much better outcome. So the outcome, you are much more responsive to that environmental intervention. And so this is where we are seeing that genes tested in Growbaby are very strongly related to fertility outcomes and improving fertility. And not forgetting about the men, that you would want to look at a couple together and looking at, say, DNA health in this scenario where the MTHFR polymorphisms are also analyzed and reported. And we see in men very interesting associations with this, also with some others that we test in DNA health, like your PON1, very strongly related to male fertility, and pesticide exposure, actually. And where we're looking at effect of nutrient intake, so this is B9 and B12, so folate and your B12 intake on semen parameters and fertility of men with MTHFR polymorphisms. Again, what they observed from this study, significantly higher frequency of the TC or TT genotypes of this polymorphism in your oligospermic, severe oligospermic, and azospermic men. The CC genotype of your other SNP, your 1298 A to C polymorphism, was significantly higher only in one state of these men with issues with semen parameters. And what they also observed was that there was a critical effect of B9 and B12 intake on decreasing of total homocysteine and improving semen parameters. So even, and this was especially seen in men with the T allele of the MTHFR 677 SNP. So again, highlighting the importance that it's not just your genotype, it does not dictate your destiny. It's looking at genotype, looking at the environment around it, and how we can significantly improve those parameters by understanding what your genotype is and what we need to do about it to improve outcomes. So highlighting here, how methylation impacts fertility immediately, but also in generations to come. And this was just another study that I wanted to, or actually another study, this is a review article that I wanted to just highlight, which I thought would be quite nice for you to read. And just again, emphasizing, I know we harp on about it quite a lot, but emphasizing the importance of the type of folate supplementation when it comes to this very sensitive time period, and really looking at your methyl folate as opposed to your folic acid. And one of the reasons around that is folic acid, because it's oxidized, it's actually a synthetic form. Even if it is converted, it's not converted fully, and a lot of it sits in your liver and can cause oxidative stress long term. So at that very high dose, you don't want that high amount of folic acid. So even though there are studies showing actually that folic acid can improve your homocysteine levels, et cetera, it's a much higher dose, and it can cause a lot of other long-term health effects that we don't want. And so, excuse me, highlighting the importance of your methylfolate over there. So that's why I included that article there, just from a practical point of view. And also showing what's really nice is if we're looking at the functional tests, what we're wanting to see is actually your red blood cell folate and not just serum folate when we're looking at folate levels, because red blood cell folate is your long-term indicator of folate levels. Serum folate, it's very short term. And so looking at red blood cell folate concentrations, that increases more after supplementation with your methylfolate as opposed to your folic acid. And that was a nice study also done. So highlighting just the importance of the forms, and again, going back to that pathway, keep it in your head where the blocks might occur and where you need to keep in mind if you have a variant there, what to do about it. So this is the genetic panel that we test and report on in Growbaby around the methylation. You'll see it's quite comprehensive in the number of SNPs that we assess. When you see a high impact factor, you know that that gene is not playing its part as well as we would like and requires that increased support. These are some of the SNPs specifically related to fertility, and there's been a lot done in these SNPs in the area of assisted reproductive technologies. What to do about it? I have mentioned quite a lot around the interventions, but in the report you get a lot more insights as well around looking at the nutrition, the food that you can start considering. So the food sources of these nutrients, looking at the supplementation that you would require from the nutrient side, and then what to do from a lifestyle side. And all of these components are important to support methylation. This is something that I'll spend more time on when there's more than an hour to go, but just highlighting the importance or I think the practical side of this, and how easy it is to take this information and put it into practice, because you're getting examples of foods, you're getting dosages of the nutrients, and you're getting real recommendations when it's over and above your baseline support required. So very important with regards to decreasing risk of all of these potential risk areas that are associated with methylation, but also for improving your fertility. In terms of your follow-up testing, some other biochemical markers you would want to look at if this area does come up as a priority for you or your patient, is looking at your methylation panel, testing for red blood cell folate, that long-term marker, methylmalonic acid, that's looking at B12, and then your homocysteine levels as well. And especially when it comes to things like preeclampsia, it's very strongly related to sort of increasing risk for that. And then also increasing risk for subfertility if it is high. Moving to the next area that I want to touch on, and this really I enjoyed doing some extra digging around, was your melatonin metabolism and your gestational diabetes. Here we're going to look at, if I can find them in my Venn diagram. So your where your gestational diabetes fits, plus your large for gestational age babies, because remember, gestational diabetes increases risk for large for gestational age, which has been associated with long-term effects, long-term disorders and diseases in the generation to come, increasing risk for certain cancers and cardiometabolic disorders. And here we're looking at this MTNR1 beta. So why gestational diabetes? I think I've already now jumped ahead of myself. Well, what it is, it's impaired glucose tolerance with the onset of first recognition during pregnancy. In terms of short-term risks, it increases adverse pregnancy outcomes, long-term risks developing type 2 diabetes in the mom, and cardiometabolic disorders, as well as increasing certain cancer risk in the offspring. it is an increasing public health concern. The rates of GDM or gestational diabetes is also increasing. And we can see that from some US data here, looking at from the early 90s to 2003, 2004, the prevalence increased from 1.9 to 4.2 with a relative increase of 122%. So this is becoming a major concern. Genetics didn't change that much, but lifestyle is changing a lot. And so we really want to target what is going on here and how can we support our genes to decrease risk and optimize health. So an area where there has been a lot of research actually into GDM, and in some very recent meta-analyses and review papers actually highlighting the importance of this area of melatonin and the SNP, and not wanting to jump ahead of myself too much, but when discussing this actually with some of the research that has been done in practice on this cohort of women who we really looked at improving their health outcomes in pregnancy, this specific SNP came out so strongly as being, I don't want to say a predictor, but it was close to really showing those women who would end up with abnormal glucose tolerance tests if the environment was not controlled. So it's a very, very strong SNP to consider. Okay, back to melatonin synthesis. So why, where melatonin is created is in your pineal gland. It is inhibited by light and stimulated by the dark. We actually require tryptophan to make melatonin as well, and some cofactors along the way, leading from tryptophan to create serotonin, and from there our melatonin is made. So really important in terms of your day-night cycle, but also showing that you're circulating, so just highlighting the importance of melatonin, not only in that day-night cycle, but also as a very potent antioxidant and in regulating blood glucose levels. So what we see, and what they've seen from research, is that we see that melatonin has been shown to have a beneficial role in controlling your blood glucose levels in both animal and human studies. So this has been found, for instance, in expression levels of your glucose transporter, your GLUT4, is reduced in animal studies where the pineal gland has been removed. And so what we see there is, obviously, if the pineal gland is removed, we don't have melatonin. And so there's a change in the expression of GLUT4, which is really important in transporting glucose. So this results in glucose intolerance and insulin resistance. But when you give these animals who've had their pineal gland removed, when you give them melatonin, it actually alleviates the glucose intolerance in those animals. So highlighting the importance of melatonin and really explaining the mechanism a little bit more. We can also see that in human studies, melatonin administration in studies that have been done in diabetics, but not in the pregnant population, but in diabetics, it's over an extended time. So it's not just a once off, but an extended period of time of the administration with melatonin. This has been shown to improve insulin sensitivity as well as inflammation status and improve redox status. So again, highlighting the importance of melatonin in actually improving or reducing risk of diabetes, gestational diabetes, and overall glucose tolerance, and improving insulin sensitivity. So the circadian manner of insulin secretion by your pancreatic beta islet cells is due to the melatonin action on the melatonin receptors, inducing a phase shift in the cells. This is where we're talking about the receptors as being very, very important. So we look at your MTNR1 beta. It's one of the melatonin receptors. We have two variants that we test for over here. And if there is a genotype that comes up, it leads to a decreased function of this receptor. So the receptor doesn't see the melatonin as well, doesn't sort of accept it as easily. And so the action of that melatonin is not as strong as it could be or as it should be. And this increases risk for gestational diabetes and large for gestational age babies. So your effects of your melatonin on insulin secretion are mediated through your melatonin receptors, your MT1 and your MT2. So your MTNR1 beta, the one that we're interested in, encodes your melatonin receptor type 1 beta, also called your MT2. This has very high affinity for your melatonin. The receptor plays an important role in mediating the reproductive and circadian actions of melatonin, and functions throughout the body to slow cell activity and promote the onset of sleep. The expression of these MT2 receptors in pancreatic beta cells proposes that their activation by melatonin might directly influence insulin or glucagon production. And we see that melatonin could stimulate glucose uptake by your GLUT4 and PPAR gamma protein expression in your trophoblasts, as we saw actually through the animal studies. And this could then, which could be attenuated by your MT2 or your melatonin receptor knockdown. So if your melatonin receptor is working poorly, you're not going to get that good glucose uptake, and you're not going to get that good expression of GLUT4 and PPAR gamma. And so we need to make sure that we have a good sensitive melatonin receptor, but we're also providing sufficient melatonin. So we see that these variants might affect pancreatic glucose sensing and insulin secretion, and thereby hypoglycemia. In some really good studies, meta-analyses, very recent ones, there's been very strong association with fasting glucose levels, moderately associated with increased risk to develop diabetes, and also glucose intolerance. And this has been found also very strongly in gestational diabetes. As we said, it's like, we don't want to call it a predictor, but it's a very strong indicator that we need to act in these women who carry the variant. So this is then based on these studies where they kept on finding this receptor coming up. This is a study I wanted to highlight with you, because again, looking at that gene environment interaction that we see. So individuals who carry the variant, so this was an intervention study actually done for gestational diabetes. So just going through some of the methods around it. It was a randomized controlled gestational diabetes prevention trial. And this is in 4 maternity hospitals done in Finland, in fact, where women with a history of GDM and now on their next pregnancy were included in this trial. So if you've got a history of gestational diabetes, there's obviously a much higher chance of recurrence. And so this group, these pregnant women, were divided into 2 groups, one with a very strong lifestyle intervention and the others just the control. And then they were also, the genotype would have been similar in the 2 groups. And what they found is individuals who carried a specific variant on this MTNR1 beta, who were undergoing intervention, had a much lower likelihood of developing gestational diabetes. So they were much more responsive to the intervention, those who carried a specific variant on MTNR1 beta. So again, showing that responsiveness and that need to intervene as early as possible. What you would also want to do, and something that you might want to consider as a biomarker for early kind of monitoring or prediction of GDM, is looking at some interesting biomarkers here. And this was a study done looking at angiotensinogen as well as melatonin. So one for preeclampsia and the other one actually for gestational diabetes. I'm focusing on the gestational diabetes here, but looking at the melatonin here, what they found here is your urinary melatonin production appears to be defective, or sorry, your activation. So your urinary levels of melatonin was lower in gestational diabetes as well as hypertension. So this ratio, angiotensinogen and melatonin, could be considered or suggested as an early biomarker for identification of gestational diabetes or hypertension. So looking at, you can test for melatonin in, say, your Dutch test. It's a very nice thing to consider for sort of those women who might be in that higher risk group. And if you're looking at the Growbaby test and you see this variant coming out, you would want to keep on doing some monitoring around some of these biomarkers. Interestingly also, and this diverts me completely, but I'll just quickly touch on it, is that the melatonin, and there were some studies showing that lower melatonin was also associated with difficulties with fertility. And so some supplements pre-conception, but to support fertility, included melatonin, and they saw better results there. During pregnancy, the studies are not concrete enough yet to support the supplementation of melatonin during pregnancy. Some say it's safe. Animal studies, it's very mixed still. And so what we're looking at in terms of supporting melatonin metabolism during pregnancy is actually how can we promote sleep and a better day-night cycle, because that, if you go into the studies, and we can dig into it a little bit more when there's more time, but really emphasizing the importance of getting someone into a very good routine in that day-night cycle to improve glucose tolerance and to improve receptor function and melatonin production. So looking at your magnesium-rich foods, things like your certain herb spices, pumpkin seeds, your cocoa, flax seeds, your chamomile has also been shown to be beneficial, also just promoting that sleep, eliminating all caffeine and stimulants. We know that a high caffeine intake has been associated with decreased fertility. And so we're also looking at just how can we, so eliminating that, but then also for during pregnancy around GDM, looking at that caffeine, ensuring a good quality prenatal, and then on top of that, your magnesium supplementation to support sleep, and then the natural melatonin production. You can also look at your tryptophan-rich foods and things like that to promote the melatonin production. And of course, your prenatal should have a good amount of Bs, which will support that methylation, and then the serotonin, and then the melatonin production that way around as well. On top of that, you could look at, it's not in here at the moment, but above and beyond baseline support, you want to ensure good sleep hygiene, avoiding stress, very high stress at night as well. So that is a brief overview of the interventions around melatonin and how it's related to GDM. Follow-up testing and monitoring, obviously making sure there's very healthy weight gain, sleep quality score you can do with your clients, checking those melatonin levels. Studies have looked at both urinary and salivary. Cardiometabolic profile, very, very important in this group. Monitoring blood pressure, that glucose tolerance, and then also checking protein. So that is just a very good monitoring if this SNP comes up. And then lastly, I wanted to focus on vitamin D and its role in preterm birth. It has many other roles, but just honing in on this area. So why preterm birth? We know complications from preterm birth is still a leading cause of neonatal death in not only third world, but first world countries, including the UK and the US. And actually preterm birth, the incidence is also on the rise. What we see beyond just the complications with preterm birth, it's obviously a longer neonatal stay, increased, as we said, increased risk of neonatal death, but a huge economic burden, increased stress psychosocially on the household, on obviously the greater community, and then for that child and that family throughout that child's lifetime, having an increased risk of disabilities, learning problems, cognitive behavioral conditions, decreased or conditions around that, increased risk for ADHD and ADD and spectrum disorders, increased risk for cardiometabolic disorders later on in life, and increased risk of that person, that mom, having a preterm baby. And so the cycle continues. So we really, again, want to step in as early as possible to decrease risk of preterm birth and improve the transgenerational health again. How does vitamin D fit in here? So in pregnancy, the fetus is totally dependent on the mother for vitamin D. So this explains the very high correlation of mother and cord blood vitamin D concentrations. And while your 25 hydroxy D crosses the placenta, your 125 OH2D does not. But that can be produced in the fetal kidneys. However, the baby is still reliant on mom's vitamin D status to be given to the baby. And we also know that there is significant inter-individual variability in serum vitamin D levels and responsiveness to vitamin D supplementation. And that is partly because of our genetic variants that we carry. I'm going to focus on the VDRs, your vitamin D receptors. These encode your vitamin D receptor, a steroid, it's a steroid hormone receptor that mediates the action of vitamin D by regulating the transcription of over 1,000 genes. So if we have sufficient vitamin D, we know that there's going to be regulation around inflammation, immunity, your bone health, and many other factors very strongly related to development. We see that vitamin D deficiency and alterations in your VDR genes have been strongly related to abnormalities in your calcium metabolism, cell proliferation, and immune function. And these variants and insufficient levels are associated with your preterm birth and small for gestational age, as well as increased risk for altered immune function in that child later on in life. And there was some really interesting studies done where we looked at where moms were measured for their vitamin D status. And if it was low, there was a much higher risk of those children having asthma. And that went on. Those were longitudinal studies done to those where those children were up to age 10. So that is how strongly that vitamin D status of the mother affects that child. Going through the pathway, briefly, we've got our sunlight, which is so important to get that exposure for vitamin D3 and that activation of vitamin D in pregnancy. If we can't get it through that, we can get it through dietary sources, but that is in a less active form that does then need to be activated. And so supplementation is generally just something that we would want to do. We get vitamin D converting to your 25 hydroxy D calcitriol and then your 125 dihydroxy calcitriol. It then gets bound and transported to your different tissues, where you can see it's pretty much important in every tissue in the body. And that is where inside the nucleus it attaches to the receptor, and that is where you then get that downstream effect of that activation of thousands of genes. So the role of vitamin D is really important in increasing or modulating insulin sensitivity, regulation of cytokines and immunoglobulin synthesis. It's looking at decreasing and modulating your renin-angiotensin system pathways, involved in regulation of calcium and phosphorus and where it needs to be. And then obviously very important in brain development and regulating the production and the signaling of neurotransmitters and the signaling or the sensitivity of a lot of neurotransmitter receptors. So important in overall body development, and obviously being a really important time period with fetal development. So we want enough vitamin D. These are the SNPs that we test for on the VDR receptor. If you carry the variant, you will see the impact factors coming up as either moderate or high, or depending on how many variants you carry, what you will see here. And just highlighting in terms of potential risk areas, there's that preterm birth risk very strongly related to that, as well as small for gestational age. And that's what you see immediately, but then long term, we can also see that asthma, increased risk for osteoporosis in that child, vitamin D deficiency, increased risk for certain cancers later on in that child as well. And that's what we want to prevent, both at preterm birth, but also risks down the line, long-term disorders. So we see that association between vitamin D plasma concentration and VDR gene variants and risk of preterm birth is very strong. But when, and there was a lovely review article, I don't know if I've got it here, but looking at supplementation was actually a systematic review of intervention studies with supplementation of vitamin D, and better birth outcomes, better fertility outcomes, decreased risk for preeclampsia, gestational diabetes, the list goes on. So you want your vitamin D levels really nice and high and good. And it's really important not to just be able to say, okay, well, I can take my prenatal or I can prescribe the prenatal and there's sufficient vitamin D in there. There have been many studies now looking at the vitamin D, even in many prenatals, is insufficient to give you the levels that you want. So that's just a note as well. And obviously, if you carry the variants, you really need to be cautious and just pedantic with intervening and providing sufficient vitamin D here and monitoring as you go. Something else that was really interesting is that another biomarker besides vitamin D that can be considered is your vitamin D binding protein. And this is almost considered as a more sensitive marker beyond vitamin D status for looking at outcomes relating to health of the mother and birth outcomes. And so very important that you could also, if you can, looking at vitamin D binding protein concentrations, and that you can test in the serum. And so if you do come out with these variants, it's quite simple, looking at your vitamin D rich foods, supplementation with vitamin D3, quite a good level, so beyond just what is considered your normal prenatal, and then looking at that UV exposure and when and how much you can get in. And that's all given to you in the Growbaby report. And what is really important here, and just to emphasize, your vitamin D related genotype provides insight to responsiveness to therapy. So flagging those who might not get the levels up as high as and quickly as we would like, and really highlighting those high risk patients again. And so in terms of follow-up testing of the vitamin D levels, your vitamin D binding protein, which can be done in serum. Just to give you an idea of what adequacy is, and that is above your 50 nanomoles per liter. And this was actually something, so this is just highlighting the study that was done by the Growbaby team in the States, and looking at specifically trying to prevent preterm birth, which they did achieve. Something that was one of the areas that came up really strongly was this vitamin D deficiency that they saw. And when you're looking at those individuals who carried a variant, it was 94% of the high-risk population that carried the variant. So you can see that they really, with correct intervention, you can get good results. And so that's what I just wanted to highlight there, with the importance of vitamin D in preterm birth. And this, I think, just a really nice, this is one of the reviews, this is the review article I was talking about. I did include it. It is open access, so you can have a read of that, and it's really nice reading to look at vitamin D in the role of fertility through pregnancy and lactation. And so overall, just an overview of where Growbaby can be used, some practical applications, diving in to the biochem, but then also how can we practically apply this information? And beyond that, what can we do is to look at not only the genetics, but your microbiome, previous triggers, previous exposures. And if we can get that all right, we aim for the goal, and we may hopefully achieve that goal of producing resilient adults with prolonged good lifespans, good fertility, and not this second scenario where if we don't have the genes, we don't have all the other tests, we don't intervene at a targeted level, we can't get that resilient phenotype and those optimal outcomes that we really are aiming for in functional medicine. So that is me, a healthy mum, healthy dad, healthy baby, and healthy world is what we're trying to achieve. If you are going to be in the London area or the UK on the 24th of September, I would love to see you in person. It's an intimate afternoon where we will be discussing practical application, test pairing with Growbaby, further insights into fertility and epigenetics, and really diving deep into this Growbaby test. And that has also been accredited with BANT CPD approved. So 4 CPD hours awarded for that. And then very exciting, we've got many delegates already signed up from all around the world to come to our Nordic conference, and Catherine has already mentioned, from 1 to 3 November in Copenhagen, with great international speakers. Dr. Carrie Jones will be speaking there on hormones from fertility to pre and post menopause. And so just a fantastic event to attend if you can as well. And if you want to register for any of these, plus the Utrecht event, please scan the QR code over here, and you can actually register for any of these events, any and all. So thank you for your time.
Thank you, Helen. A few questions came up about the dad. So let's say we have the perfect scenario. We have parents, and they want to have a healthy pregnancy and healthy conception. Now we know what to do with the mom, but should we also do a DNA health with the mom and a Growbaby and a DNA or core? And then the dad should at least do health, if not core.
I would say definitely if you can look at doing, I would say it depends on how much they're able to do. But in this space, I would say maybe for mum, and especially in the fertility space, Growbaby, and then looking at the hormones side of the genetics as well. I think that would be a very nice detailed insight into fertility, if that is specifically the area that you're wanting to do. So I would say Growbaby and the estrogen and the hormones on the genetics for the mom. On the dad, I would say a DNA core would be really good, because, and then probably the hormone side as well. So the estrogen and hormone side on the dad side as well. And I think from a genetic side, you could probably get a lot of insight from that. We will eventually, hopefully, get into sort of a Growbaby dad. But I would say DNA core provides a lot of the insights, especially on the methylation. And that is something I would want to, I have planned a few detailed sessions on that for the dad on fertility, but it's at this event. But looking at so your methylation, your MTHFR SNPs, and further, they've actually done some RCTs for them, and looking at success, and then also looking at, which is reported in DNA core, is your lipid metabolism SNPs, things like ApoE and PON1, which play a very important role in fertility in dad, and the exogenous stresses, especially like your pesticides. So, and then your oxidative stress for dad. So I think from a genetics testing point of view, I would say that would be your DNA core would really actually cover it really nicely, plus the hormone side. So genetics of your estrogen metabolism and all of that would just give that further insight. And I would say maybe for mom and dad, they could do that as an extra one. And then looking at some of the functional tests based on, so yeah, the organic acids, your Dutch could be very useful alongside that.
So there's been a few questions, and I know we're running over time, but it's so exciting. And I think first of all, there's been some really nice clinical comments with like success stories in their clinic, and also Keegan, he's like very supportive in regards to he's sharing some of his own story in the chat. So thank you for sharing that, and it's very, very encouraging to read. So then vitamin D. So I was like, so one question was what is optimal levels of vitamin D at preconception, and then you had a slide, and then you said above 50. In Denmark, 50 to 150 is the reference range. And I know there's this U-shaped curve that's in regards to mortality, not pregnancy. And that says like there's least mortality around 125. So, I mean, I think there's broad discussions on where we should be on vitamin D levels in blood. And just because we have optimal levels in blood, if we then have VDR receptors, then we might even need even higher blood levels based on the interpretation of the genetics around D vitamin. Can you comment on that a little bit, maybe? Or
Yes, so
that could be a
I
whole other-
almost wanted to just do my whole presentation on vitamin D, because there was, and I'll try and share it actually with the presentation. There's an article looking at actually mentioning how vitamin D is, it's good as a marker, but especially during pregnancy, because not all of, so what you're testing for isn't always your whole picture. And so you've got your sort of your free and your bound vitamin D. And so it's difficult to actually get good insight just looking at vitamin D as a mark on its own for are you at sufficient levels. And so that's why, and Catherine, as you say, there's such a broad range, and it's because of this huge inter-individual variability of how it attaches to that receptor and how it works post, like what just the normal level is. So for some individuals, yes, I mean, 50, we definitely want to go high, and I agree with you, 100 around there is a better sweet spot. But actually not just looking at vitamin D, but looking at vitamin D binding protein. This article is now recommending that might be a much more sensitive marker for health in terms of fertility outcomes and healthy pregnancy outcomes. So don't know if I've just made it more complicated, but it's to really aim for something that is in the higher range. What they mentioned from a toxic level, I mean, it was very high, so it would be very difficult to get, and that's where you just want to continue monitoring. But actually measuring things like vitamin B binding protein would be much better.
Super. So I think that's I have to go to the beach now, just saying.
Yeah. I'm waiting for the sun.
But Helen, as usual, this was just like so nice, and I hope some of you are gonna join Helen in London, and if nothing else, then just join us in wherever we have events and where Helen is showing up. Thank you all for joining us for this long and staying tuned. Take care, everyone. Bye, and thank you, Helen.
Thank you. Bye.
If you do not already have an account with us, please contact our team and we will assist you. If you found this webinar helpful, please like and subscribe to our channel.